Hello everyone. As promised, today we will explore the 4 phases of clinical research. Generally, clinical trials is the stage of drug discovery where a promising compound finally begins its journey in humans. We will explore each phase in order. Phase 1, which answers the question, is this compound safe? Phase 2, does it work in patients? Phase 3, is it better than what we already have? And finally, phase 4, which is about answering the question, what happens after approval? You will see how each phase builds on the previous one, how decisions are made, what questions are answered, and how evidence is gathered step by step until the regulators and healthcare systems are confident enough to bring a new medication to the world. Let's start with phase 1, which answers the question, is this compound safe or not? Phase 1 is where the real journey of a new drug in humans begins. And it's often described as the moment of truth, because up till this point, everything we have known about this new compound has come from the lab experiments and animal studies. We have seen a promise, we have seen some data, but none of that guarantees how it will behave in the human body. So the question we now ask is very simple, but also very critical. Is it safe? And because this is the first time the drug is introduced into human, the phase marks the transition from preclinical testing to clinical evaluation. And the objective here is not to find out if the drug cures the disease. That will come later. In this phase, our goal is purely about safety, tolerability, and understanding the body response to this compound. So who participates in phase 1 clinical trials? Typically, phase 1 trials involve a small group, usually 20 to 100 participants. Most of the time, they are healthy volunteers rather than patients, unless the drug is considered too toxic for healthy individuals, as in the case of oncology medications or HIV drugs, where actual patients participate even at this early stage. Of course, each volunteer gives informed consent and is also closely monitored in controlled clinical settings, often at specialized phase 1 research units. Their participation is vital, and every single observation, even if it is a mild headache or a change in the appetite, is carefully recorded. As I mentioned earlier, researchers are not yet looking for therapeutic benefit. Instead, they are conducting what we know as first-in-human studies, designed mainly to answer fundamental questions like, how does the drug behave inside the human body? How quickly it is absorbed into the bloodstream? How long does it stay there? How it is metabolized and eventually excreted? Then, these studies focus on what we understand and know as pharmacokinetics and pharmacodynamics, which is the science of how the body affects the drug and how the drug affects the body. Participants may receive a single dose or a multiple escalating doses, depending on the type of the design. In single ascending dose studies, which are called SAD studies, small groups receive gradually higher doses, helping the investigators to determine the maximum dose that can be given safely. Later, multiple ascending dose studies, which is called MAD studies, may test repeated dosing to see how the body handles ongoing exposure to the compound. So safety here is a central question. Throughout phase 1, safety monitoring is constant. whether we are talking about vital signs, blood tests, ECGs, and sometimes imaging, are used to detect even subtle changes in the body. Every side effect, whether mild or severe, is carefully documented and analyzed. The objective is to identify the dose range that is considered safe, the point where the drug has an acceptable safety profile without causing any harm. This becomes the foundation of all subsequent trials. Let's take an example to make it clear. Imagine a new anti-inflammatory drug that performed remarkably well in animal studies. In phase 1, researchers give the first small dose, perhaps 1/10 only of what was safe in animals. They give this small dose, to a group of healthy volunteers. They monitor heart rates, liver enzymes, kidney function, and more. If everything looks fine, the next group receives a slightly higher dose, and so on. By the end of phase 1, researchers can confidently say, at these doses, the drug is safe and well tolerated in humans. If unexpected toxicity appears during phase 1, the compound may be stopped altogether, saving time, cost, and most importantly, preventing harm in a larger group. If the drug passes phase 1 successfully, it means we have crossed a crucial threshold, which is the proof of safety in humans. We now know how the drug behaves in the body and how the body handles it. We also know how it can be dosed safely and what the potential side effects might be. This knowledge becomes the green light to move to phase 2. And in phase 2, we will shift our focus from the question, is it safe, to another question, does it work? Does it actually work? Exploring whether the drug can actually deliver clinical benefits to the patients with the disease. So here we go, phase 2 clinical trials. And as I mentioned, it answers the question, does the compound actually work in patients? Because after establishing safety in healthy volunteers during phase 1, the spotlight now moves to the people who truly matter, patients living with the disease the drug is originally meant to treat. And phase 2 is the moment when science meets reality. Now, the central question here is, does it actually work? Which marks another transition from safety to efficacy. And this is the first time the drug is tested in real patients who have the target condition. It is an important transition because now we are looking at whether all those years of research, early testing, can really translate into actual clinical benefit. In other words, we are asking, does this drug improve symptoms? Does it slow the progression of the disease? Does it change measurable outcomes like, for example, tumor size, blood pressure, or blood sugar levels, depending on the therapy area? For example, in oncology, phase 2 might measure tumor response rate, disease progression, or patient survival trends. While in diabetes, it might measure the change in hemoglobin A1C, and so on. Every disease area has its own definition of what working means. But the core goal remains the same: to find evidence of clinical benefit in patients. That's all what we want in phase 2. And phase 2 is often divided into two sub-stages, phase 2A and phase 2B, each serving a clear purpose. Phase 2A is about early signs of success, and we call this type of studies proof of concept studies. It's usually conducted on a small number of patients, sometimes few dozen or up to 100 participants. The main purpose of type 2A studies is to look for signals of efficacy. Does the drug appear to make a difference compared to placebo or the existing treatment? For example, if a new compound aims to reduce inflammation in rheumatoid arthritis, phase 2A might measure the reductions in joint swelling or pain scores or other markers after few weeks of the treatment. On the other hand, phase 2B are those finding studies. So once early signs of effectiveness in phase 2A are seen, phase 2B can begin. And here researchers fine-tune the details, particularly the optimal dose. The objective here is to find the sweet spot, the dose that provides maximum benefit with minimal side effects. Because too low a dose might make the drug ineffective, and too high a dose might cause unexpected toxicity. In phase 2, several dosing regimens are compared head to head, often using randomized and controlled designs to ensure the results are reliable. This is where statistics, careful patient selection, and clinical judgment all play major roles. Phase 2 typically involves few hundreds of patients, generally between 100 and 300, depending on the disease prevalence and study design. These trials are often multi-centered, conducted across multiple hospitals, research centers to ensure diverse patient representation. Remember that unlike phase 1, these participants in phase 2 trials are patients with the condition, meaning they are often already on some form of treatment, or they may have some limited conditions. That's why ethical oversight, informed consent, and patient safety monitoring are especially important during phase 2 clinical trials. Let me also share with you the fact that some experts call phase 2 the valley of death in drug development. Why? Because this is where most experimental drugs fail, unfortunately. A compound may have looked safe in healthy volunteers, but when tested in real patients with the disease, the study looks completely different and the story can completely change. That is why it's so tough. Why tough? For three main reasons. Diseases in the real life are multifactorial, as we all know, and they vary greatly between different patients. Also, don't forget the placebo effect can sometimes blur the efficacy or early efficacy signals. And also, the safety margins may narrow when drugs are given over longer durations or alongside with other medications. In fact, statistically, more than 50% of drug candidates entering phase 2 never make it to phase 3. Let's make this practical with another example. Imagine a new oncology drug that showed excellent tumor reduction in animal studies and was well tolerated in phase 1 human studies. Now, in phase 2, the same drug will be given to 200 patients with advanced lung cancer. Researchers will track the tumor size, survival time, and side effects. If most patients show stable or shrinking tumors with acceptable safety, that's a green light to proceed. But if the results are mixed, perhaps the drug works only in small subset or probably cause unexpected toxicity. It might be modified or even completely discontinued. So studies in phase 2 provides the make or break insights that define the future of the drug. By the end of phase 2, the development team aims to have 1. Evidence of clinical efficacy that the drug shows measurable benefit for patients. And 2. A safe and effective dose range for future trials. And finally, preliminary data on the side effects that could shape the patient selection and monitoring in the next phases. If these conditions are met, the drug seems to receive its ticket to phase 3, where larger, more definitive trials will determine whether it truly deserves approval. In phase 3 clinical trials, the central question is this compound better than what we already have? By the time a new drug reaches phase 3, it has already come a long way. It has proven to be safe in humans, the proof of safety in phase 1, and have shown promising signs of efficacy in patients, phase 2. Now it's time to answer the ultimate question. That will determine whether it can become a real-world medicine or not. Is it better than what we already have This is the phase where all years of discovery, investment, and scientific effort are put to the final test. It's also the most expensive, most complex, and most decisive stage of drug development. Phase 3 trials involve hundreds to thousands of patients, often across multiple hospitals, cities, and even countries. And the goal here is to ensure that results are reliable, representative, and applicable to real clinical practice. Because diseases can behave differently across different populations based on genetics, environments, diet, or healthcare systems. Conducting studies in several countries actually helps researchers to confirm that the new drug performs consistently anywhere. This global scale gives phase 3 trials tremendous credibility, and it's also why they are considered the gold standards despite they are time consuming and costly and often taking several years to complete, but they are still the gold standard studies. Phase 3 trials are designed to be objective and unbiased. And to achieve this, they use what's called the randomized, double-blind, controlled design. Randomized means patients are randomly assigned to receive either the new medication or the new compound or a comparator, usually an existing treatment or a placebo. This ensures fairness and prevents selection bias. Double-blind means neither the patients nor the investigators know who is receiving which treatment, and this eliminates subconscious influence or expectation. Controlled means there is always a comparison group. Without this, you cannot tell whether the new treatment truly makes a difference. This design ensures that any differences in outcomes can confidently be attributed to the drug itself, not to happen by chance or to be caused by any bias. The objectives of phase 3 are clear, but very demanding. First, to prove whether the new drug works better than the current standard treatments. 2, to confirm its safety profile in much larger and more diverse group of patients. 3, to establish a clear benefit-risk ratio showing that the benefits outweighs the potential risks. For example, a new oncology drug is being tested. Researchers here in phase 3 might compare tumor shrinkage rates, overall survival, or quality of the outcomes between the new therapy and the existing standard of care. The drug must demonstrate either superior efficacy, improved safety, or sometimes both, depending on its intended role in the therapy. Phase 3 generates the key evidence that determines whether the drug can be approved by regulatory authorities like the FDA or the EMEA or by other health authorities in different countries. Every dosage, side effect, and clinical outcome from phase 3 clinical trials becomes a part of the regulatory submission package, which is known as the new drug application or marketing authorization application. If the data show that the drug is effective, safe, and clinically meaningful, regulators may approve it for use. If not, even after years of work, the project may end here. This is why pharmaceutical companies and sponsors treat phase 3 as the make or break moment in the entire drug development journey. Let's take another example. Consider a new cancer therapy designed to target a specific mutation. In phase 3, the company runs a global study with 2,000 patients across, let's say, 20 countries, comparing the new treatment to the best available standard of care. Results show that the patients on the new drug live on an average 6 months longer, with fewer severe side effects. That's the kind of evidence regulators and clinicians look for. It is not about statistical significance alone. It's about real-world meaningful benefit for patients. Let me add here that logistically it's complex, coordinating thousands of participants, data collection systems, ethical approval worldwide. It's also financially intense and sometimes accounting for 70% of the total drug development costs. Moreover, unexpected challenges can arise. For example, the drug might not outperform the standard therapy, or rare side effects may appear only when large numbers of patients are treated, or data inconsistent between regions may raise concerns and questions. Despite all this, when phase 3 is successful, it represents one of the greatest milestones in medicine. It's the point where hope becomes a potential reality. In the phase 3, if data confirms that the drug provides a significant clinical benefit and has acceptable safety profile and offers an improvement over the existing treatments, then the sponsor proceed to submit regulatory review and approval. And once approved, it can be prescribed to patients and often enters phase 4 clinical trials where real-world data continue to be collected. And ladies and gentlemen, here we are. Phase 4 clinical trials, which is meant to answer the questions, what happens after approval? This is the final phase of clinical research, and it is known as post-marketing surveillance or phase 4 clinical trials. And it is exactly where science steps beyond the controlled environment of clinical settings. At this stage, the medicine is now being prescribed to thousands, sometimes even millions of patients across diverse populations, healthcare systems, and geographical regions. And while the earlier phases of clinical research told us that the drug can work and it is safe, phase 4 studies tells us how it really works in everyday life. The earlier phases, even the large phase 3 studies, cannot capture everything for many reasons. One, because they are conducted under strict conditions with careful selected participants. Remember the inclusion and exclusion criteria and close monitoring from the investigators. But the real world is completely different. Patients have multiple diseases, comorbid conditions, they take other medication, they live varied lifestyles. So now the focus of phase 4 clinical trials has shifted to understanding how the drug behaves outside the perfect clinical setting or the laboratory setting. In simple words, phase 4 is all about long-term safety, performance, and optimization. The key questions that this phase answers are: 1. Are there rare or long-term side effects that didn't appear in the earlier phases? How does the drug perform in special populations, like, for example, elderly, children, pregnant females, who may not have been already part of the earlier studies, phase 2 and 3? How does the drug interact with other commonly used medication? Does its real-world effectiveness match the clinical trial results? These questions help to refine our understanding of the medicines and ensure that its benefits continue to outweigh its risks. And phase 4 clinical studies, they come in different forms. Some are observational studies, where data is collected from patient registries, electronic health records, or even insurance database. And the objective here is to monitor outcomes over time. Other studies in phase 4 are real-world clinical studies designed to compare effectiveness across different treatment groups, or surveillance programs that collect safety reports from physicians and patients. This is where pharmacovigilance, which is the science of detecting, assessing, and preventing adverse drug effects, plays a critical role. The pharmaceutical industry have dedicated teams and systems to collect and analyze safety reports globally. If any unexpected risk emerges, they can quickly update the product labeling, issue safety communications, or even temporarily withdraw the drug if needed. One of the most valuable outputs of phase 4 is real-world evidence data gathered from actual clinical practice rather than the controlled trials, as we mentioned. Regulators, payers, and healthcare providers now use this evidence to make decisions about reimbursement, coverage, guidelines, and treatment optimization. For example, such real-world data helps regulators confirm that the medicine performs safely in different diverse populations. It helps also physicians to understand how the drug fits into evolving treatment protocols. And it helps companies, the industry, to identify new opportunities, such as exploring additional indications, adjusting dosing schedules, or developing new formulations for specific patient groups. In oncology, for example, a drug that was initially approved for one type of cancer might later be found effective in another cancer based on the real-world data, leading to expanded approvals. And in many regions, phase 4 is not an option, it's mandatory. Certain health authorities require companies to conduct post-approval clinical studies or risk management plans as a condition of registration. This is especially common in therapeutic areas like oncology, immunology, vaccines, chronic diseases in general, where the long-term safety and population variability are major concerns. For example, the European Medicine Agency often mandates post-marketing studies under the post-authorization safety study or the post-authorization efficacy study frameworks. On the other hand, the US FDA can also issue phase 4 commitments as part of the new drug application approval, meaning that ongoing data collection is legally required after the launch of the product or the medicine. Also, several countries around the Middle East and Asia, their regulatory agencies now require real-world safety and utilization data within the first few years after registration. And this is to maintain, or a condition to maintain, market authorization. This ensures that the medicine's benefit-risk balance remains favorable as new populations begin to use it. If post-marketing data at any time reveal any concerns, regulators, as we mentioned, may take actions to update the label or to provide or restrict the indications, or in rare cases suspend the marketing authorization until further evidence is provided. Let me also add here that phase 4 is not a responsibility of the pharmaceutical industry alone. These studies are considered shared responsibility among all key players in the healthcare ecosystem. So pharmaceutical companies will continue to collect and report safety data. Regulators will evaluate the ongoing evidence and enforce post-marketing commitments. On the other hand, healthcare providers will monitor and report adverse events. Even patients play a vital role by communicating their experiences and outcomes. Together, this ecosystem ensures that the medicine remains safe, effective, and trusted long after it reaches the market. So, ladies and gentlemen, as we have seen, bringing a new drug to life is not a single event. It's a long, meticulous process built on questions, evidence, and accountability. Remember, phase 1 clinical studies asked, is this compound safe, proving that the compound can be used in humans? While phase 2 asked, does it work in patients, showing early signs of benefit? Phase 3 asked, is it better than what we have already? And this phase delivers the clinical data that convince regulators and physicians to approve it. And finally, phase 4, which continues the story, ensuring that the medicine remains safe, effective, and trusted in the real world. Each phase represents a step forward in science, but also in responsibility. That's the big picture of clinical research, and it's what ensures that innovation reaches patients safely, responsibly, and with hope. Thank you very much for joining this tutorial, and see you next time.